Hey everyone, today we're going to talk about the central control of ventilation. This is a topic that I'm going to split up into 3 videos, where the first video I'm going to talk about central control ventilation, the second video I'll talk about peripheral control ventilation, and then in the third video we will put it all together so that we can understand ventilation in more depth. So control of ventilation and control of many body systems is a sensor integrator effector model. It's also known as a negative feedback system. So meaning that you have a sensor that will sense something, usually a biological marker. It will be fed into an integrator, which is a central control area, usually in the brain, which will then lead to an effector, which is usually a muscle group, to then lead to a change that is then sensed and then fed back in, and the cycle repeats. For central control ventilation, we'll talk about the central sensors. And we'll also talk about the integrator, which is the brain stem, particularly the pons and the medulla. So let's just orientate ourselves a little bit here. If we have the brain side on, the pons is located here and the medulla here. So it's in the brain stem. Now if we zoom in and we're looking front on now, the pons is superior to the medulla. And I'm going to just split it in half as well for the midline when we start talking about ventral and dorsal medulla. So for these different respiratory nuclei, I'm going to use blue for inspiration and red for expiration. Now some of them do have functions in both, but it's just easier to talk about it in respect to inspiratory and expiratory nuclei. So with the pons, the first one is the aponeustic center. And the aponeustic center is located in the lower pons, and it prevents overexpansion of the lungs, and its activity is associated with prolongation of the ramping inspiratory potentials of the dorsal respiratory group, which I'll talk about later. It gets its name as it refers to the inspiratory gasps observed when the pons is sectioned above this area. Now just above the aponeustic centers is the pneumataxic center, and this is a center that's associated with inhibition for the respiratory ramping activity of the dorsal respiratory group, and this overall reduces the tidal volume and increases the respiratory rate. Now if we move down to our medulla, on the left we have the dorsal medulla, so posterior medulla, and on the right we have ventral medulla, which is the anterior portion of the medulla. So in the dorsal, I'm just going to also draw the fourth ventricle just up here as well. And this is just posterior to the medulla. So the first nuclei is the nucleus tractus solitarius, marked with NTS. And this is a nuclei that's important in the dorsal respiratory group, also known as DRG. And it's important because the cranial nerves 9 and 10 also join up around here. So it receives feedback from these nerves. And its motor branches, or its axons, are actually ipsilateral as well, so they travel directly down from this nucleus. It mainly consists of inspiratory neurons, as I mentioned before, that control the diaphragm and are responsible for normal tidal inspiration. It's mainly concerned with timing of the respiratory cycle. Now on the right we have the ventral respiratory group. And this is made up of the Botzinger complex and the pre-Botzinger complex. So the Botzinger complex is important for expiration, while the pre-Botzinger complex is also known as the pacemaker and is responsible for inspiration or timing the inspiration. Essentially, it commences the inspiration. And they're active during normal tidal breathing, while the nucleus ambiguus and the nucleus paraambiguus and the nucleus retroambiguus are more associated with active breathing, so involved when you need to take more active breaths in and active breaths out, because expiration is generally passive. In regards to their tracks, the Botzinger complex crosses the midline, so contralateral, and it inhibits the inspiratory neurons of the nucleus tractus solitarius and the pathways descending down. The nucleus ambiguus is ipsilateral, so its tracks run in the same side, and the nucleus paraambiguus crosses the midline, and so does the nucleus retroambiguus as well. Your ventral respiratory group can also be broken up into a rostral and caudal portion, with the rostral portion being your nucleus ambiguus and the caudal portion being nucleus paraambiguus and nucleus retroambiguus. So these are essentially the integrators, but the sensor is located around the midline in the medulla, and it's generally in this sort of area, and it's the central chemoreceptors which sense hydrogen ions, which I'll talk about in more detail as we progress through this video. Now let's just put it together, and I've oversimplified it a little bit here just to make it easier to understand. But if we take our pons, we've got the apneuistic area, which modulates ramping of inspiration, and our pneumotaxic area, which inhibits ramping. And then in our medulla, we've got our dorsal respiratory group and our ventral respiratory group. And remember, our ventral respiratory group is made up of our pre-Botzinger pacemaker cells, which usually stimulate the start of inspiration. And our dorsal respiratory group has our nucleus tractus solitarius, which is also involved in inspiration. So if we take our pre-Botzinger pacemaker cells, they essentially commence the inspiration, and then when we need a bit more active inspiratory effort, we might engage our nucleus paraambiguus, but our nucleus ambiguus is otherwise engaged. And it will inhibit expiration, which remember is for our Botzinger complex and our nucleus retroambiguus. And there's this interplay and feedback between expiration and inspiratory nuclei as they feed back to each other. Now, our nucleus tractus solitarius and our dorsal respiratory group, they will promote the inspiratory effort that's generated from the nucleus ambiguus and nucleus paraambiguus. And these overall lead to inspiratory neurons, which will then stimulate, for example, the phrenic nerve, which innervates the diaphragm for inspiration. And the nucleus tractus solitarius will also promote these inspiratory neurons as well. And when you need active inspiratory effort, you will engage your accessory muscles of respiration as well. So this will lead to lung stretch as the lungs fill up with air. And this stretch will be sensed by, for example, the vagus nerve and its innervation into the lungs, which, as we mentioned, it's got efferents that go all the way up to nearby the nucleus tractus solitarius and the dorsal respiratory group. And then they will inhibit the inspiratory effort. So this inhibition means that your inspiratory neurons will reduce the amount of firing that they have, meaning that the expiratory neurons will then fire more, which leads to your expiration. And generally it's passive. So generally just by inhibiting the inspiratory neurons, you will naturally have some expiration. But then if you inhibit them more and you need to expire faster and have a faster respiratory rate, you can then also engage your active muscles of respiration as well. But these expiratory nerves will lead to inhibition of lung stretch as the lungs deflate, which will then reduce the amount of inhibition from the lung stretching on the DRG, and the cycle repeats, and you'll get this repeating cycle of lungs inflating, inhibiting inspiration, leading to expiration. Expiration then will cause less inhibition on inspiration, which then leads to inspiration taking effect again. Now again, we've went into the integration and how these higher centers control breathing, but I haven't really talked about what actually makes us breathe. Now in your textbooks, many people talk about CO2 as the stimulant to breathe. And that is in a sense correct. It's CO2 in the blood that will, if we got a rise in CO2, it will lead to increased respiration. And generally for every millimeter of mercury rise in partial pressure of CO2, you expect about your minute ventilation to increase about 2 to 3 liters per minute. But that's not the whole story. I mentioned before that hydrogen ions stimulate breathing. And I'm going to show you how that works and why hydrogen ions are the reason that we breathe or stimulate our breathing. But it's also closely linked to our partial pressure of CO2 in the blood. So in this picture here, we've got blood on the left. We've got our blood-brain barrier as the thick line next to blood. Then we have CSF, and then another line, which is that brain parenchyma, and the central chemoreceptors sitting within the brain parenchyma. Now, hydrogen ions themselves can't actually cross this blood-brain barrier. If the concentration is very high, eventually some will leak across the blood-brain barrier, or if the blood-brain barrier doesn't work, for example, in meningitis and other conditions where it's broken down, then yes, hydrogen ions can stimulate breathing. But it's actually the CO2 that can easily cross the blood-brain barrier because it easily dissolves and diffuses across. So the CO2 will bind with water that sits within the CSF, creating carbonic acid, which will then become bicarb and hydrogen as it dissociates. And it's this dissociated hydrogen ions that then cross over into the brain parenchyma and stimulate our respiratory centers. Now, there's no clear place that it stimulates. I don't think anyone is actually certain, but it's hypothesized possibly the ventral respiratory group, as it's close to that area and it's involved with inspiration and expiration. But I don't think this is exactly clear. So when in doubt, I probably would just leave it blank and just say stimulates respiratory centers. So this is why the hydrogen is actually closely linked to the partial pressure of CO2. So it's the CO2 in the blood, the partial pressure of CO2 to be more exact, that crosses the blood-brain barrier and leads to increased hydrogen ions in the CSF. An important feature of CSF is that it's actually a bad buffer, meaning that small changes in CO2 that cross over into the CSF lead to changes in hydrogen ions, which is important because it allows us to then be quite sensitive to CO2. But the problem with this is that if your CO2 is chronically elevated, you'll get some compensation that happens within the CSF as more bicarb is made to try and maintain a normal pH of the CSF. And then this decreases the sensitivity to partial pressure CO2 in the blood. And then these people become dependent on hypoxic drive, which the brain actually directly doesn't sense hypoxia. It's the peripheral chemoreceptors that can sense hypoxia and drive respiration. And this is what I'll talk about in the next video, where we will look more into the peripheral receptors and how they function and how they stimulate respiration, as it's quite different to the central chemoreceptors and the central functions or controls of ventilation.